Je sais pas si ça va.
Yes.
Ouais.
Yeah, it's working. Okay, everybody, sorry, we are going to start.
Yes. So you say a few words before, or?
So today we are receiving David, David and Alice Bueno.
Okay. Okay, so good morning, everyone. So it's my pleasure to welcome Davide. I don't know how to pronounce it. Thambiturai. Okay,
Yeah.
the Italian way.
Ouais.
Yeah.
Ouais, en fait, le nom, c'est sur l'enquête, parce que je suis italien.
Okay. So Davide is a fish biologist and a comparative physiologist who is currently working at University of Glasgow. And he completed actually his PhD also in the same university, where he explored how commercial fishing practices can drive evolutionary changes in fish behavior and physiology. And he was using zebrafish as a model species for his work. Since then, he has been doing several postdoctoral projects across UK, France, and also beyond. I believe also in South America, isn't it?
Yeah, in Brazil and Indonesia, a little bit.
Yeah, and he did some studies from studying basically salmon migration in Scottish rivers to examining movement ecology and physiological condition in tropical tuna in the Indian Ocean. So he has been collaborating closely with UNR Marbeck, where he has done a lot of work. Basically, he has worked on research topics such as the effect of fish aggregating devices on tuna or oxygen limitation on Mediterranean fish. And he generally combines fish field and laboratory approaches, integrating telemetry, physiology, and advanced data analysis to understand how fish respond and adapt to environmental and human pressures. And today he will be sharing his work on selection and evolution in the Anthropocene and looking at it from a fishery perspective. So please, Davide, the floor is yours.
Okay, thank you, Bruno. So I should just specify that I am an affiliate researcher with Glasgow University. I'm not currently there, so I'm still based in Montpellier for now. Okay, so I wanted to start off with this quote, which I think is a really poignant quote. So, man, the hunter, has become a mechanized techno-beast, a highly efficient predator. So this is a quote that I read at the start of my PhD in 2015, and it really stuck with me. And I think the sentiment of this quote is it really captures the talk and the subject area. So we, as predators, have surpassed natural predators in our efficiency, in what we can remove from the wild. And what we should think about, an important aspect of that, is thinking about the evolutionary implications of this. Okay, so humans, if we think about the Earth's development and history in 12 hours, this is an analogy I came across relatively recently. Eukaryotes came around 5:30 hours after hour zero, let's say. And then fishes came around 10 hours 14 into that, so quite late on. But humans, if we think about how long we have been on Earth, we've been on Earth about a second, if we look at that in that 12-hour scale. And within that tiny little amount of time, we've had a huge impact on Earth and the organisms that live on Earth. So that 1 second still equates to around 300,000 years, which Homo sapiens have been around. And in that time, we've had a lot of impacts, but they've been concentrated in the last 200 years or so. So with the Industrial Revolution, etc. And so this has not only been on the direct harvest of species, so such as hunting and fishing, there's been declines in the species, but also whole-scale environmental change, such as agriculture, urbanization, etc. So these impacts not only influence population dynamics of species, so we see a decrease in the number of individuals, but they have driven evolutionary change, and that is something that we should consider going forwards. So evolutionary change that occurs in short time scales, either decades or a few generations, is called contemporary evolution. And human activity, because of the strong selection regime that we can impose, has actually driven quite a lot of this. So some famous examples include the Anopheles mosquito, for instance, that builds up resistance to insecticides very quickly. So there's been lots of work on this in Africa to try to get rid of these because they are malaria carriers, but quite quickly they build up resistance to these insecticides. Another is these beautiful elephants with really huge tusks. These are called tuskers in Africa. And these tusker elephants have been declining since the start of the century due to selective hunting for ivory. So because they are more prized, they're really big tusks, people have been hunting them specifically and they've been removing them from the population. And therefore now there's a lot less of them. And I think one of the most kind of examples used in evolutionary teachings is the peppered moth. So the peppered moth was a moth, or is a moth in Europe during the Industrial Revolution. Whereas before the Industrial Revolution, there were quite even numbers of moths, of this like white moth and a dark moth. With the Industrial Revolution, with soot and dust laying on surfaces, they were predated upon, the white moths, and therefore there was a shift towards a darker moth. What is interesting was a quick shift, and after, when the Industrial Revolution started finishing and there was less dust, it went back to kind of even numbers between those two moths. So all three of these examples are examples where physiological or morphological phenotypes have been targeted and have been removed perhaps from the population, have led to rapid evolution, so contemporary evolution. In natural systems, predation is amongst the strongest drivers of evolution and also contemporary evolution. So a seminal study in this kind of context is this work by Resnick in the 90s. And what they did, they took Trinidadian guppies in Trinidad from areas where there was only one type of predator. So this predator that targeted adult fish, so adult guppies. And then they transplanted individuals to some areas that had juvenile targeting predators. And they did this to try to test life history and evolution theory. And what they saw is that quite quickly, in the areas within 11 years, the areas where the adults were being targeted, the fish were displaying early maturation, higher reproductive effort, and smaller offspring. Whilst in the juvenile targeting areas, they saw later maturation, lower reproductive effort, and fewer offspring. So these results really demonstrated natural selection and proved some of those life history theories that they were trying to identify. So in this case, we're talking about natural predation. So we're talking about relatively diffuse selection, perhaps, and weaker selection differentials. So if we contrast this to human predation, such as fishing, which we'll be talking about, human predation can be considered, not in every instance, but can be considered much stronger and much more directional. So that's why it's important to think about the evolutionary implications of this. So to kind of illustrate that point, I just wanted to bring your attention to this statistic. So this was a great paper by Darimont et al. In Science in 2015. And what they showed is that for one fish caught by a natural predator in the oceans, they estimated around 14 fish are caught by commercial fishing. So it's a huge, huge difference in mortality, right? So, you know, assuming that fishing isn't random, there's got to be some evolutionary potential there. And given that this pressure is much greater, perhaps it's quicker also. The questions we should be posing ourselves, if this is the true mortality of commercial fishing in comparison to natural mortality, what are the implications of this? What are the phenotypes that are being captured? How fast is evolution? And all of these evolutionary implications are important. Okay, so going back to basics, if we have a genotype, which is the genetic makeup of an organism, we then have a phenotype, which is the observable, expressed, basically, organism as a function of the environment and its genes. And assuming a very, very simple system, let's say with some cows, and you will find out when I'm talking about cows later. We remove the largest cows, okay, and then theoretically, if this is a heritable trait, what we would see is like smaller cows in the next generations, etc. So why am I talking about cows, when I say? Because the first time that fishing was actually related to evolution was in the context of cows, okay? So this is a quote from 1902 from Cloudsley Rutter. He was a fish biologist working in North America. So I'm going to read it out. A large fish is worth more on the markets than a small fish, but so are large cattle worth more on the market than small cattle, yet a stock raiser would never think of selling his fine cattle and keeping only the runts to breed from. So the runts in English basically are the ugly small cows. Okay. And so what he was referring to in this case is the commercial fishing that used to go on in North America. So using these gill nets, so they're kind of like curtains that hang around in the water column. And salmon migrated to go into the rivers. And as they migrated, these fish were caught. And these nets particularly targeted the larger fish because they would get hung, the larger fish as they go through the net, but the smaller fish would swim through and go on to reproduce in the upper streams, right? So you're saying, okay, a farmer would never keep his ugly cows to breed, but we're doing the same to the fish, okay? And so he was talking about size selection, basically. And despite this comparison, it was until much later that this kind of evolutionary effect of fishing was considered. Okay, so what I'm going to show you now, I hope that the resolution in here is going to be good, is a video. Okay, and it lasts about 2 minutes, 1 minute 50. And it's of fishing, okay? And what I would like you to think about is kind of the selective pressures that might be occurring. It's quite dramatic, and I think the sound for this in the room is going to be coming out of here. What do you think swimming in front of that? Okay. Right, so sorry about the resolution here. So I hope you enjoyed that. So that was the great David Attenborough commentating on that short video. But that video showed a very common type of fishing, which is trawling, chalutage en français. And the main message that I wanted to get across there is that fishing of that type and other types of fishing isn't random. Often we're targeting certain phenotypes within a population of fish, okay? So historically, we've centered trying to understand what the effect is on size and growth, but more and more, we're starting to see evidence regarding performance. So other trait categories, such as performance, for instance, swimming capacity. So we can think about selection on swimming capacity within the context of that trawl. As the trawl is going forward, fish that can swim better are going to be able to get away within a population and swim, and fish that are slower perhaps are going to be entrained and go into the net. So there's a selection there. So other kind of categories, we think about morphology, physiology, life history, and behavior. So one of them is very important, I think, is physiology and metabolic rate. Metabolic rate is kind of a basal trait, let's say, and could influence upper level traits such as swimming physiology because they're very closely related. Okay. Right, so the field in which we try to understand the effects of fishing on evolution is called fishing-induced evolution or fisheries-induced evolution. So to begin with, there was a major focus on size selectivity, as I said, and this was until the 1930s. So from 1900 to 1930, there wasn't really much work done, and then between that and the 2000s, there was some experimental work done, but globally, the field really exploded around 2000 with Richard Law. After 2000, one of the seminal pieces of work is this work by Conover and Monk in 2002. And what they did to try to prove basically the effect of fishing on life history, they did some common garden experiment. And they took this fish here, Aterina, it's an Atlantic silverside from the wild, put them in the lab in different groups and replicate groups, etc. And then they subjected them simply to size selection within those groups. So at the end of each generation, they removed some fish of a certain size, right? And so this figure here shows growth rate on the y-axis and generation on the x-axis. And what you can see is that very quickly, within 4 generations, you have these very different responses to selection. So the circles in this image are indicative of the removal of small fish. So the larger ones are able to breed. Here's a removal of large ones. And you can see this kind of very disparate response to that selection. And here in the middle, we see a response to random selection. So if the fishing was random, this is what you would expect, right? And this was amazing work. They proved that fish can have an influence, but it was relatively limited in its scope because they were really just basically removing the fish, such as like selective breeding, basically, in this case. But what it did prove is that also there was recovery issues after they stopped. It took a while for the fish to recover in the growth rate. Okay, so FIE, fishing-induced evolution, consideration of traits other than just that size and that growth took a while longer. So these are just some of the examples, but behavior, so traits related to behavior. Quinn, for instance, looked at the run timing and the migration of Atlantic salmon and North American salmon. And what they showed is that because of selective fishing in this figure here, on specific peak migration days, there was a delay in migration as the years went on. And it was important that this was controlled for environmental factors, etc. So obviously it wasn't some sort of environmental thing influencing the migration of the fish. Diaz-Pauli in 2015 looked at 2 different types of gears, so trawl, so like you saw in the video, and traps, and looked at how these 2 different types of gears can select on a shy, bold, or timidity kind of complex. And then later on, we look at effects of physiology. So Redpath looked at how angling influenced metabolic rate in bass in different lakes. So lakes that were angled had a lot, there was a lot lower metabolic rate than the other lakes in which angling didn't occur. And then Killen in 2015 also looked at used simulation to look at the influence of trawling on capture in trawls. And this figure here shows exactly that. So we have the total time that the fish spent inside the net, and then metabolic rate, standard metabolic rate, which is basically the energy needed for an organism or a fish to survive. And what we see is a negative relationship. So the lower the metabolic rate, the higher the time that they spent inside the net. And what the link there was is probably related to swimming. So SMR was found to be related to swim in this case. So it was what the point here is that it was quite a while until we considered other traits other than growth and size. Okay, so just a little bit more about metabolic rate. As I said, it's a very important trait. So in fishes, metabolic rate can be simplified to three main components. So we have standard metabolic rate, which is the aerobic metabolism needed to keep a fish alive, basically. Then we have maximum metabolic rate, which is the maximum aerobic metabolism that they can use. And then between those two is what is called aerobic scope. So this is basically the envelope of energy I have to do different things, okay? So we can split that into different components, so digestion, growth, activity, so activities such as swimming. And we can also fish can shift that envelope to suit their needs, okay? And sometimes, such as in the case, for instance, of the trawling, all their activity might be towards doing one thing, so swimming away from a trawl, okay? And we also have anaerobic metabolism. So this is energy they can muster that is not using oxygen. So in fish, a lot of times when they're escaping trawls, what they do is glide, burst and glide, and this type of movement is associated to anaerobic capacity. And there could be selection on this component of the metabolism also. The last thing I want to say regarding this is that stressor can influence the way that fish can utilize their energy. And so in some cases, selection perhaps by fishing could influence the way that they also respond to climate change in some ways. So in fish, metabolism is often measured with respirometry. So this is a respirometry setup. The reason I mention this is because I've kind of presented some data and I don't want to do it every experiment. But in essence, what we do is we put the fish inside a chamber and we measure the oxygen consumption. And that gives us an estimate of the metabolic rate. So measuring selection in actual fisheries is quite difficult in the wild. So often what is done in this type of work is to utilize simulation approaches. So what are simulation approaches? So if you can imagine this graphic here is just a simplified diagram, but it's an arena in which we use a model species such as guppies or zebrafish or other species which can kind of simulate what's going on in real life in the larger fish species. And then we subject them to specific selection processes, perhaps. And as I was saying, the bulk of work to try to understand selection has been done on this. In the wild, the bulk of work has been done looking at the response to selection. What this allows us to do is to have fine environmental control of the selection process and understand and tease apart very specific processes, okay? And so here you can see there's a camera, for instance, monitoring, and then we might be monitoring what the fish are doing, etc. So in 2015, I started my PhD at the University of Glasgow within an ERC project, Fish. And my role within the project was really to try to develop simulations to try to understand how fishing can select on physiology, behavior, and performance of fish. And so this is the whole team, postdocs, lecturers, etc. Okay, so the first thing that we did was to try to establish a framework by which fisheries can select on physiology, morphology, and performance. So we came up with this. In essence, the main thing that's important is this capture continuum idea. So the capture continuum is an idea related to how different types of gears and the way they work will select for different fish. So we have this active side. So active gears, fishing gears, are things like the trawl that you saw. And we have the passive, which is something like a trap or someone that's angling without moving, basically, right? And just a little bit more about that. Active gears pursue fish like the trawl. They capture fish as they fatigue or as the fish turn within the net. and may preferentially capture fish with, for instance, low swimming ability or low aerobic capacity. On the other hand, we have the passive gears, which are static and rely on the fish finding the gear, and they may preferentially capture fish, for instance, that are more active. So if theoretically SMR having a higher metabolic cost requires you to look for more food, you might be more active, you might be caught more often, theoretically. Yeah, and then we kind of assumed made assumptions about what traits might be selected for on each side of this continuum. Okay, so now I'm going to present several kind of experiments. The general structure will be there's a question, and the title of the question won't change, but then I'll go through the methods and the kind of results. So one of the first things that we were interested in also understanding how group behavior can influence selection, right? So fish are often social, they hang around in groups, okay? From a natural selection perspective, being in groups is often thought of as beneficial. So being in a group allows you to look out for predators, because more fish looking out for predators allows you to find food quicker. So these are positive things generally, right? But if that's the case, what are the dynamics between fishing and fish schools, right? Is it the same? Is it a positive thing to be in a group? So, as I was saying, using some simulations, I used zebrafish, and I basically set up, we set up an arena that would allow us to track the fish and look at the response going to these traps. So we designed these traps to have GoPros on them. So basically all the fish were individually marked, and we could see the fish that were going into the trap and out of the trap, and we could quantify different metrics. So the important thing, we're interested in a focal fish within the group, not all of the fish, okay? And the metrics that we might be related to vulnerability to capture are entry latency, the time that the fish are spending in the trap, and the number of entries that they're having in the trap. Okay, so fish were trialed in 4 different groups. So individuals, so the focal fish, as I was saying, the purple fish here, +2 shoalmates, +4 shoalmates, and +6 shoalmates. And many replicas of these groups were done, and fish were never trialed twice. They were only trialed ever once, okay? So here's a video of one of those trials. Okay, so the fish are released, you can see. It's a very simple model. There's nothing in here. There's not many things. These are 3 plastic plants, basically. But what you'll see is that the fish are hanging around, and they're trying to eat this spot. I don't know what it was, but it was obviously interesting. And then what you'll see is at a certain point, one of the fish will go into the trap. And this is a key moment, because as soon as that happens, you'll see what happens with the other fish. So as soon as one fish goes in, which in this case has gone in, the others are attracted to that fish because they're like, oh, what's going on? He's there, he's moving around, he's making a commotion. And so the others are going in as well. Okay, so this kind of a shoal behavior might not actually be very good for fishing. And so you'll see all the basically they're all going in now. Okay, so here are some of the results from that study. So there's the 3 metrics. So we have entry latency, time in the trap, and entrances on the panel on the figure on the right-hand side. So first of all, entry latency. On the x-axis, we have the group size. So individual on the left, +2, +4, and +6. So what we see is that the individuals, this, remember, it's not the group, it's the individual within the group, is more likely to spend, to go in quicker into the trap when he's in a group. So that's the first thing. Time that they're spending in the trap is also much higher for the 2 groups with 4 individuals and 6 individuals. So the time they're actually spending in the trap can equate to vulnerability to fishing, because the more you're spending in the trap, the more likely is probability when you're going to be, the trap is going to be pulled up and you're going to be caught. And the number of entrances, the number of entrances is also higher for those higher groups. Okay. So that's that. And then we have, so in the top right, we have some survival curves that were fitted. So if we hypothetically think about that 6 treatment as a population, we can see that the decrease in that population is a lot stronger than the decrease in the other population. I also measured metabolic rate in these fish, and there was some evidence of metabolic rate effects, though there was an interaction between the size of the group and metabolic rate. It wasn't extremely clear in this case. So that work was later picked up, and much more work has been on top of this. And kind of this idea that grouping can influence, can be selected for in fisheries. So this work by Guerra et al., and they showed at the population level that that fishing could actually select, and something that has evolved to benefit fish in a normal natural scenario could actually be making them more vulnerable globally and changing the kind of shoal cohesion and shoal evolution. And this work here also by Bragaia, kind of theoretically also expanding on that and looking at lots of different types of fishing. Okay, so group behavior could be selected for, not very good. Okay, so next, we have another question on how do active, so going back to trawling, so this is an active, and passive fishing techniques select for fish shape. Okay, so I was interested in understanding how morphology was being selected for. Morphology reflects how a fish interacts with its environment. It's really important for feeding strategy, swimming efficiency, habitat use. So it could have a lot of implications, selection on shape. Oh, so here is, so what I did, in addition to the simulations I showed you earlier for trapping, my colleagues and I created simulations for trawling. So these are zebrafish within a simulated trawl. So what happens is that the fish are in a recirculatory flume. They are basically put in the chamber, and then they are allowed sometimes to interact with the fishing gear. And then we quantify basically the outcome of the fishing. So for each fish, we build a vulnerability index. So we did one for trapping and one for trawling. For trawling, basically, if you think about, this is a recirculatory flume, the fish are swimming against the current, and they have many different options. They can either stay in front of the net, they can go behind the net, they can go in the net, and they can also go basically so far in the net, and they go into this triangle that you see here. And this triangle allows them to be caught but not be in the current. So that's the worst case scenario. So if you're in a real trawl and you were in there, you're basically done for, pretty much. So this is the kind of that index I was talking about. So we constructed indices for all of the fish that were trialed in trawling with these different outcomes, and all of the fish were trialed 3 times. So we did this for trawls and for traps. So I'm going to show you a video of a trap scenario for this. Okay, so the fish are released, and similarly to earlier, the fish are allowed to interact with the trap. So the traps are baited. So this is just as it would be in real life. And yeah, so they're given a certain amount of time to explore this arena with the traps in there. And at the end, remember that we are quantifying at the individual level the vulnerability of each fish. Okay. And so you can see that one is eating there is the clever one, maybe. I'm not sure. But again, we have this kind of, oh, what's going on? The others are interested to see what that fish is doing. Okay, so after that, what we use geometric morphometrics to look at the shape of the fish. Okay, so this is a technique often used in evolutionary studies to look at the shape of things, accounting for allometry, so different sizes, accounting for different size. And what we do is we look at homologous landmarks on the fish, so very specific landmarks, and sliding landmarks in this case also. And we do that across the kind of fish, and we do this for all of the fish. So we've built up their vulnerability, we know what their vulnerability to fishing is, and now we're looking at their shape. Okay, so what we see in the results is there is convergent selection in shape between the gear types. So both trawls and traps seem to favor catching the kind of skinnier fish globally. Okay, so you can see these two. So the top is trap, the top figure here, and the bottom is trawl. And on the two, the meshes at the top, there's the high vulnerability individuals, and here as well. And on the two lower thresholds, the green is the low vulnerability individuals. So shallow fish were more vulnerable generally. But what we hypothesize is potentially like the pathways leading to that selection are different. In swimming, often what we saw is that there's a deeper caudal peduncle and a kind of narrower head, and what that is indicative of is swimming performance. So in general, in fish, when they have a big caudal peduncle, they're very good swimmers. So we theorized that the selection was happening because the skinnier ones were being caught because they're not so good at swimming, and the bigger ones were good at swimming. On the other hand, for the traps, what we looked at was scale mass index. So this is an index of condition in the fish. And we saw that the condition in the fish was lower for the fish that were caught in both methods. So there could be some interaction there for trawling as well. But we assumed that the fish that were being caught in the traps were hungrier, and their internal state was such that they would want to go and feed more in the trap. Okay, so globally, it seems that different gear types, at least in this case, in this simulation, seem to have induced similar selective pressures on the morphology, so the shape of the fish. Fish escaping the trawl showed a deeper caudal peduncle and narrower snout associated to swimming. And so what I want to make clear here is that this selection only represents the selection that happens just before the fish are caught, not at wider scales. So we have to think about selection perhaps at wider scale also. So that's something I'll go back to. Okay, so next question. How does an additional stressor influence selection in trawl? So hypoxia, low environmental oxygen, low in water, how does that influence my ability to be captured by a trawl or the selection regimes? So oxygen's concentrations in marine areas have been declining for the past 50 years and are continuing to decline. And we know that swimmer performance is influenced by oxygen availability, because if I can't use oxygen, I can't swim as fast, right? So my aerobic scope diminishes. How does it modulate selection then in trawls? So I'm not going to show you. Oh, obviously. Okay, so again, I did trawling simulations within this recirculatory fins, just as I showed you earlier. But in this case, we have different treatment conditions. So we have normal oxygen level, and we have low oxygen level. And we try to identify what the effect is. So for low oxygen, we were considering a 35% air saturation level, so relatively moderate hypoxia. And the fish were trialed multiple times, so 3 times in hypoxia and 3 times in normoxia. And basically, I was interested in two metrics, so binary capture, whether they're not their capture, slightly different from earlier, and the time that they spent in the net. So you can see in the pictures here, so you can see there's a stone, and the way that we induced the hypoxia was to put nitrogen in water, so it replaces oxygen. And here is a recirculatory stream with the trawl, okay? Okay, so here's the fish swimming. Okay, well, you saw that, but basically here they were trying to hypoxia and normoxia. I'm going to go a little bit quicker because I want to get through some stuff. Okay, so the first thing we see is that swimming performance influences capture. So fish that are swimming faster are the ones that are getting away. So this kind of reinforces the idea that if you're swimming badly, you're probably going to get caught more. Okay, so there is selection there. And also we see that normoxia and hypoxia have a really big impact on swimming. So if we look at the figure in the middle here, this is UCRIT, so critical swimming speed. In normoxia, so the blue spots, the fish are around 80 centimeters per second, whilst in hypoxia around 70 centimeters per second. Okay, so quite a decline in swimming capacity. But the most important thing, I think, from this study is this third panel, right? So what we see is that variance, which is basically what is needed for selection to occur, collapses under a hypoxic scenario. So the top panel, we see that critical swimming speed in normoxia is always quite wide. So the population start, they're below 70 centimeters up to 95 centimeters within my population in normal conditions. Whilst when we look at hypoxia, there's basically very little variance, so between 60 and 70. So what does that mean? That means that under a hypoxic scenario, fish that might have been escaping or being caught, they're no longer doing that. Basically, nearly all the fish are being caught, okay? So this particular stressor seems to have an influence on the selection process. Okay, so yeah, so hypoxia has a modulating effect on swimming performance, most likely through metabolism. The capture rate in this particular study increased around 7-fold, and what is most crucial is that the variance collapses and also the repeatability, the individual repeatability of fish collapses. In the sense that in normoxia, if you're a fast swimmer, you're going to swim fast every time. But in hypoxia, sometimes you'll swim faster, sometimes you'll swim slower. It kind of goes out the window if you're fast or slow. That's what we saw. And so stressors may drastically alter fishery selection. So just to prove that this, well, to kind of think about what does this mean in the wild. So I came across this and kind of other examples, but this is an interesting quote from a paper. Several days of unusually large catches of sole preceded a period of 0 catches as severe hypoxia developed. So they saw a huge increase in capture, and the most likely result is that fish were not being able to get away from the trawl as they were fished. Okay, so the next study, I concentrated on parasitism, and I went to visit Sandra Binning at the University of Montreal, and Sandra is a fish physiologist working a lot with parasites. I took advantage of this really cool system in near Montreal, so an hour north of Montreal, at the biology station of the Laurentides, of 3 different lakes. And these 3 different lakes had these fish called pumpkin seed sunfish, and these fish had really different densities of this parasite. And the parasite, I don't know what these spots are there, but this parasite here, you can see this fish is a parasite fish, infected fish, with basically these parasites that have metasaccharida on top of the skin of the fish. And they are a trematode that one of its kind of later life stages is this metasaccharida stage. And so what we did is, because the lakes differed very much in the density of these parasites, we thought, okay, let's think about how parasites influence selection. I've looked at selection in lots of different things of fisheries, right? Okay, so again, I did respirometry to measure the metabolic rate of the fish and how the parasite interacted with the metabolism. I also did some trapping simulations differing with individuals different in infection. So similarly to earlier when I had 4 groups but different numbers of fish, in this case they were always tested with 6 fish, but in some cases there were 6 uninfected fish, 2 infected, 4 infected, and 6 infected. Okay, and then again metrics were binary capture and latency to enter and time in the trap. Okay. So similar to earlier, the fish are in a much bigger arena. So this is over 2 meters, I think, this arena. And these are very, very interesting fish because they often stay static like that. So they move and they stay still. But what is also interesting here, you'll note that I have little spots on their head. And then I put little colored pieces of paper so I could automatically track them. So they all had this little spot on their head. So you can see one goes in, and again, the others are attracted to it, and this is a reoccurring theme, right? Okay, so results regarding that. So first of all, metabolic rate positively relates to parasite infection. So we see as more parasites are on the fish, the cost of living for the fish is increasing, right? So what you would expect, perhaps, if that's the case, then if I need to eat more, if my cost is going up, I need to eat more, so I'm going to be trapped more. But that's not really what we saw. We didn't see that the effect of parasite infection on latency to enter trap, so how long it took them to get in. There was some effect of time spent in trap, but this was a bit confusing. What we saw is that the time that fish were spending in traps was highest for the most the groups that had the most similar individuals. So either 6 infected or 6 uninfected. spend the most time in trap. And we assume that this is some sort of conformity in behavior that arises perhaps when the group is conformed, they kind of behave similarly. Yeah, so the kind of what came out of that is that black spot certainly increases SMR, the base cost of living, though there wasn't really clear or strong relationship with the selection. Again, only tested the final moments. You'll see why I'm talking about this, but final moments before captures. And it would be interesting to see how other parasites, because there's so many different types of parasites, how selection is influenced in other contexts. Okay, so kind of going towards the end now. So this is something that arose a few years ago, but what I was thinking about is we looked at the effect of these stresses that kind of has an influence on the selection regimes that fishing may impose. So I really concentrated on that idea. So how does fishing select in this changing environment? So one of the things that can influence selection perhaps is environmental conditions. So if it's hot, if it's cold, if it's raining, if it's not raining, whatever the environmental condition, oxygen levels, etc. Another is fish condition. So the internal state of the fish will influence whether it's captured or not. If it's really hungry, maybe it's more likely to be caught in the traps, etc. And finally is the capture process itself. Is the capture process always the same or is it changing? And so to try to unite these things, we can think about the kind of the stressors or the things that might influence fishing, right? So one of them is technology. So at the start, well, let's say 200 years ago, this is what fishing looked like. Me on a canoe, throwing a line in and trying to catch a fish. And now we have these trawlers that are 100 meters long with nets that have, the size of 7 jumbo jets into kind of size. So obviously selection must have changed from then to now. The weather, if it's good, if it's wavy, it might change the way that fish behave and therefore selection. Parasites, again, I mean, there was kind of weak evidence for parasite. I think it's worth looking at that a little bit more. Pollution, so there could be effect on metabolism, there could be effect on physiology that could influence how fish are selected by fishing. And then population density also can have big effects. There's some recent work that has come out in PNAS that relates to this on the selection regimes and on evolution. So all of these things kind of can have an effect. And what we did so the question we should be posing ourselves, do fisheries function in a shifting selective landscape? And how can they function? What are the environmental influence that influence selection? At what scale does fishery selection occur? Does it occur just at a small scale, like in the trap examples, or at the larger scale? And is selection is selection on some trait less likely to be flexible? So is it always, so for instance, if we're thinking about mesh size is always pretty much going to be selecting whatever the environmental conditions. The fish are not going to be able to get through that if they're too big, okay? And so last year with a collaborator in Finland, Anna Kuparinen, we came up with this kind of review and contextualize this shifting selective landscape. And so just to go through this figure here, so fishing selection has to happen through a series of steps. Okay, so in here we describe 4 different steps. So the fish and the fisher, the fish must overlap the habitat. The fishing must overlap the habitat of the fish in global terms. So same sort of region, Gulf of Leon, let's say. Okay, so if any of these steps, the fish falls out of the capture, it's not going to be selected for, it's not going to be captured. So the second step is the fish must encounter the gear at a kind of a more localized scale. First step is the fish will detect the gear, either escape or not escape. And then lastly is the fish is in proximity of the gear, the fish will go in the gear or not in the gear, right? And the spatial scale, this is an important aspect here, is either very large, so we're talking about kilometers, or very small, centimeters. So the selection scale is changing also. And we kind of coined this these 2 terms called flexible fishery selection. So this is selection that is much more prone to be influenced by the environment that the fish finds itself or any other kind of influence. And then this rigid fishery selection. So this is selection that will always happen regardless of anything that's happening around, perhaps. And so we also hypothesized the kind of traits that are being selected at the two different ends of this. So perhaps behavior is much more flexible. So selection on behavior might be, very prone to be disrupted, whilst selection on morphology, such as size that we were talking about earlier, is less likely to be disrupted. Okay, so just to finally, so FIE is a unique example of contemporary evolution. So I think, given the strength of fishing, it's a really good place to look at for contemporary evolution. At the beginning, FIE was related to just size and growth, but we're starting to see a lot more being influenced by fishing in lots of different traits. So we're basically running a system with fishing, perhaps similar to domestication in that we're selecting certain fish, but we're making the fish better suited to the conditions established by humans. Perhaps we're working against natural selection that is occurring in the sea. We don't really know. There's still a lot of work to be done on that. And so one of the things that I think Bruno's looking at is we could be unknowingly eroding natural genetic variation within populations. And this has been, there's a lot of evidence for this. I haven't covered it here, but there's quite a bit of evidence looking at genetic evidence in the wild. And that's it. Thank you very much for your time. Wait 2 seconds.
You can talk here.
Okay, so I think there's irrefutable, pretty strong evidence, very strong, that fishing selects for size. Okay, so that's not something, I'm not arguing that that's not happening, or no one is arguing that. What I guess the sort of work that I've shown is that there might be a lot more that we're selecting for. And okay, yeah, fishing selecting for bigger fish when they go into the net, because for instance, the very historical view is that the bigger fish can't go through the net and they're caught. But not only this, is that fishers are always interested in the bigger fish because they bring more money to them if they catch the bigger fish. But there could be a selection that happens a lot of different scales that precludes selection within the net. And so we should also think about this, because okay, maybe I'm catching the bigger fish of the fish that are exposed to the fishing, but at large scales, I'm not actually, some of the fish are already being selected, so I'm already biasing my selection somehow. I hope that's clear.
Yeah, but yeah, my question was also that the concern also comes from the thinking that the largest and the smartest and most experienced fishes are removed, but that may not be the case, like always. So I thought
Yeah, I guess it might not always.
not always correct.
I guess it might not always be the case that the largest ones are removed, especially if there are some sort of if there is some selection happening before you get to that stage. Yeah.
Yes, go ahead.
Do I need it?
I think we
I
have.
think that's it.
Can they hear? They can hear.
Yeah.
I'm just gonna try and talk loudly.
I'll repeat it.
Thanks for your talk. I was wondering, you showed experiments where you were targeting mainly one trait at a time.
Mhm.
And some of the traits that you listed are correlated
Yeah.
genetically or phenotypically, like
Yeah.
body size and metabolism, for
Yeah.
instance, but also behavior if you have a broader pace of life perspective, for instance. So I was wondering if you were able to identify what type of selection pressures were direct or indirect, depending on these correlations, and even whether the correlations may evolve because of selection pressure on one trait. And
Yeah. So thank you for the question. So there's been quite a bit of work looking at correlated selection, and certainly correlated selection happens. So for instance, between metabolism and behavior, that's something that can definitely happen. It's not something that we tested specifically, correlations within our case.
but did you have the data to do it? Did you have fish that were phenotyped for different kind of traits where you could look at least the phenotypic correlation?
Yeah, you could look at the phenotypic correlation. I guess the difficulty is over generations. We didn't have the capacity. Well, we didn't have the capacity to do that. So it would be interesting to establish the heritabilities within that generation and then look at the next generation. And it's just something that's much tougher to do. But I think it's a very interesting line of inquiry, and it's just something I haven't done. But correlated selection is something that has been discussed widely within the subject area, and it's something very important because obviously, if we're selecting for size, as you said, and we're having an effect on lots of different things, then I think the situation is very, very difficult to understand, basically, because of this. Yeah.
Thank you, David. I was in that scheme that you had like selective pressure selection at the center and the three forces. You know, I could easily replace this with the word vulnerability. Like everything in a way you presented. But I could also, you know, like revisit that from the perspective of what would make fish vulnerable to fishing.
Yeah. Oh, yeah. I mean, vulnerability. I guess selection is. Is it the same as vulnerability? I mean, vulnerability has to be there for selection to happen, right? So the fish, the phenotype, whatever we're targeting, has to be vulnerable to the fishing for it to happen. And as I was saying earlier, I guess that vulnerability is prone to shifting depending on the condition and the context that the fish finds itself in. And that's why sometimes you might see very strong selectivity in one scenario, but very weak selectivity in another scenario.
I think if I may,
Yeah.
I think I'm asking this because you know I was thinking you said there is selective pressure, but at the same time everything you presented in a way has been going on for quite some time. So I was wondering, for instance, you know, like whether actually you are implying that, and then I'm expecting to find less social or less group.
Yeah.
Or to find,
Okay.
you know, like more faster swimmers
Yeah.
out there.
Okay,
Right?
so
I was confused
yeah.
about, you know, like the let's say the cause of the effect.
Yeah. So let's say the fastest swimmer. So something that is never really been picked up, but has never been published officially. I think it was an ICES report in the Baltic. It was really interesting. So what they saw is that it was two Russian guys studying this. So I'm concentrating on the swimming for now. But what they saw is that after a certain amount of years, I can't remember how many years, maybe 15 years or 20 years of trawling, what they saw is that the vertebrate, there was an increased vertebral count in the fish that were being targeted. And what they associated this is because the fish were actually, you know, the ones that were being captured were the slower fish, and the ones that weren't being captured were the faster fish. And that's why there was this increase in vertebral count. So you can get relatively quick morphological changes. The difficulty is it's kind of like saying, you know, selection function like this. It's just very difficult to say because things are so dynamic. And, you know, I think one of the things that I discussed in that paper, the kind of review paper at the end, is that it's so dynamic. You know, fishing technology is changing all the time. So, you know, selection would be really working towards one specific phenotype, but then something drastic may happen that will change selection totally. So I think that's something that is, we have to like keep on top of basically if we're trying to understand it. Okay. So sorry, I just put on the screen some actual genetic evidence. You know, there's various different things, and there's also various work supporting, you know, selection and evolution in fisheries, and also saying, you know, that there isn't any effect. So it's very, very confused, I think, globally, the field.
Right.
I think that's it.
Thank you, thank you very much.
Okay, thank you. Alrighty. Should I shut down the Zoom?
Yes, you can.
Okay.